go through how about the method to describe you the methods that we've been developing over the last 4 or 5 years that have been collected and put together in a single toolbox, which is called FRIT, which is a Python package for functional connectivity analysis and group level statistics, especially designed for neurophysiological data.
You've basically covered so many different methods that can be used on a data set, so many different ideas on how to arrive at measures of interactions. So in practical terms, if you are confronted with a new data set, how would you go about? What would be your strategy or roadmap to basically get a first intuition?
Yeah, very good question, Michael, because this is, especially for someone who's never done functional connectivity analysis, all this may appear extremely abstract and extremely, where shall I start from? What the hell? I have my data, I have a keyboard, then what should I type on my keyboard? This is crucial. And I hope really that the school that you've organized will allow to do this, because it's really important to go down to the basics. Basically, this was actually, so thanks not to myself, so I don't take any merits for this, but mainly from Etienne Combrisson, who is this guy in the picture here, that actually taught me a lot of things about how to organize the code, organize the data, organize the workflows, as you're saying, in a structured way, so that you can build from core functions and then scripts and then workflows and organize them in a simple way that allows everyone, I hope, even with a very little experience, to go from your data to the end result, which is a figure. I mean, we don't have any kind of visualization tools, but to the end result, which is like a statistical significant effect at the network level. So now we'll go through the core functions, and then I will end up with what we call the workflows, which actually answer directly your question, Michael, which is how do I start from the data and how to organize the data such as that I can go through the whole pipelines of analysis. So
Cool.
basically, I suggest you to have a look at this. So that actually the Python toolbox is present in this GitHub, which contains the code. And then there is a documentation, which is on this web page, which contains the basic formalism, basic notions of information theory, and then lots of examples and tutorials of how to analyze your data. So now we'll go through the core functions. So FRIT is organized in different levels or hierarchical levels of complexity. And the core functions are, of course, stored into the FRIT core function. And all of what I told you until now, so all the maths, all the mutual information, entropy, and so on, is contained in a single Python code, which is called GCMI ND. GCMI because it stands for Gaussian coupled mutual information, ND and dimension, because it allows you to work with not only with matrices, or not only with between two vectors, for example, between X and Y as in vectors, but it's generalized for tensors, which simplifies, also speeds up all the calculations. This function here is based on the code developed originally by Robin Ince, which is the first author in this paper 2016 on human brain mapping that introduced the Gaussian coupled mutual information, which is copied here, that it was adapted and for 1D variable, multi-value variable, but not to tensors. And in these functions, you will see all the definitions that we've looked into, such as you will find functions to calculate, definitions to calculate the entropy, conditional entropy, mutual information, Gaussian coupled mutual information between discrete variables, continuous variables, basically all the core functions that you may need to analyze your data. if you are familiar with the Python and you are kind of geek style as us, this is the single and only function you may need to use if you don't want to go through all the statistics and so on. You may just need use this function here for your analysis if you want to just pick up some basic ideas from this function without going to the full workflow that we propose in the later steps. If you are more advanced, this is actually the function that I would suggest you to have a look and use in your project. Basically, as I mentioned earlier, this function allows you to calculate, say, the emission information between two continuous variables, such as you see here. Imagine you have two signals. It allows you, or between n, like 100 brane regions, it allows you to calculate the functional connectivity between these two time series in a static manner. So this is an example of a single pair, but you may want to do this between, if you have X and Y, which are two matrices, it will allow you to calculate all the pairs in very rapidly. And it will allow you to do this also in a dynamic fashion by doing a sliding windows approach to calculate what we call the dynamic functional connectivity. Or even simpler, you may use this function to calculate the conditional emission information or the Granger causality, or what we defined earlier as the transfer entropy, by calculating, say, the emission information between the past of region Y in red and the present of region 2, conditioned on the past of itself. And there is one function which allows you to calculate in a static manner, so by taking the whole time window, or in a dynamic manner. So as I was mentioning, you may start from this and you may write yourself these functions starting from the lower level core functions. Or if you want to do these functions, you may use a set of functions that we added and in the functional connectivity directory. So the connectivity directory, which is stored into the frit/con directory, uses the core functions to calculate and it contains code to calculate some of these metrics, which are functional connectivity metrics. And there are several of them. On the other hand, as I mentioned earlier, the advantage of using information theory is that you can use the same core function. So we always use the same building block, which is the GCMI_ND function for all the different functional connectivity measures. For example, you can use the con_DFC, so dynamic functional connectivity, to estimate the functional connectivity in a dynamic manner between two time series or in a network, okay? So the input is going to be the time series organized in time, trials, and then regions, so in a tensor. And this function will output very easily, in a very quick manner, the dynamic functional connectivity in a time-resolved manner. Or maybe you may want to calculate the transfer entropy across trials. So you may want to use the connectivity_TE for transfer entropy, or the covariance-based Granger causality if you want to calculate it over time, and so on. These are some kind of higher level functions that you may want to use directly on your data. On the other hand, if you, little parenthesis, also based on the very first question of Michael, how do I infer the values depending on my data? So as I mentioned earlier, there are different ways to estimate the entropy values and the emission information values depending on the type of the data and depending on the assumptions that you can make. If you can calculate your data and assume and transform it to be Gaussian, okay, in that case, this will be what we use in most of the time. Your estimator will be a Gaussian coupler emission formation. On the other hand, over the years, we realized that people may want to use different or custom-based techniques, such as binning or like kernel-based methods, to be sensitive to more complex relations. And so we decided to add a new component into Frit, which is contained into the directory called estimator, which contains different types of way to estimate the mutual information from the data. So you can use either the binning methods or the GCMI methods, and then you may also develop some custom-based methods if you want to link it to some external software such as scikit-learn or so on. So depending on your level of expertise, you may want to use either standard methods or different estimation methods. Then the second step is going to be, in both cases, either if you're using the core functions or if you're using everything which is built into Frit, you may need to, at the end of your project, to say, okay, I'm finding some functional connectivity which is changing over our experimental manipulation, but does it really make sense? Because I'm testing 100, or say I'm testing 500 neurons, 500 brain regions, and then there is like 5% that is coming up as like with a very strong value. But does it really make sense? Is it really statistically significant? So in that case, of course, the answer is do the stats and do it in a correct way. As I mentioned earlier, you should use tools that correct for biases due to multiple comparisons, and also in what we are using to generate new distribution permutation-based statistics. So there is a directory into Frit which contains all the parametric and non-parametric tools to do statistics. The MCP is the multiple comparison problem code, so set of code that they do this. The stats non-parametric contains all the code to do the non-parametric analysis, the permutation-based analysis. So these are the two that we use most of the time. On the other hand, there is also another tool part that is implementing parametric measure that in case you may want to use it, so you may also want to explore it. So everything put together, this is what the shape of your workflow looks like. So you have your data on your computer, you want to have final result, which is a set of p-values which are corrected for multiple comparisons. What do I do in between? So in this scheme, you may need to choose different methods depending on the type of statistics and group level statistics that you may want to do. So if X is the neuronal data, which is normally arranged in a 3D tensor, which one dimension is time, the other dimension is trials, and the third dimension is the number of brain regions or the number of neurons. This is normally how we organize the data in a simple manner. In that case, you have a bifurcation here. You may want to ask yourself, but am I analyzing like a random effect? Say I have data from multiple participants, so I may want to use a random effect analysis. Or shall I'm analyzing, say, the same brain region, say the same LFP electrodes from a Utah array, which has not moved over the days, okay? So I may want to ask myself, is there any kind of fixed effect which is common, in which you may want to concatenate different sessions for different recordings of, say, of a subject. In that case, you may want to calculate on the fixed effect across your different recordings. So imagine that in this simpler case, what you end up calculating is the true effect, okay, which is, say, the mutual information between the functional connectivity in between your brain, between your neurons and the experimental condition. and then the permuted mutual information by randomizing the order of your trials. Okay, so this will be the only step that you need to do before going to the second step, which is the calculation of the statistical inference. So the first block here is only the estimation of the data that you put into the next step, which is the statistical inference. On the other hand, here at the beginning, if you need to combine your data, which is composed of multiple subjects, okay, in that case, you may want to have like a model of the group data across participants. Say you have 20 participants. So in this case, you may need you need to calculate the effect size, so the emission information and the permutation per each subject, or per monkey, or per animal, and so on. And then at the next level, you need to calculate like a group level model. Normally, you do like you have a Gaussian model of your data. So in this case, you will end up calculating, say, true effect, like a t-value, a mean over the population, and the true effect, and then the true effect due to permutations. So this will be the output that you would put into the second step, which is the statistical analysis. The goal of the second block is to calculate the effect size and the permutations at the population level. And here again, you have two solutions that you may use depending on your goal. Either you may want to calculate the effect size for each sample in your data, say for each time point, for example. And in this case, you may need to use what is called a test-wise statistics, okay? Or you may want to use tools which allow you to calculate clusters of statistical significance. And in that case, you will use the cluster-based methods. The notion of cluster, so the tools that we are using that have been developed over in early beginning 2005, 2010 by the team developing Field-Trip. And the concept of cluster is very general. A cluster can be in time if you have a univariate signal, so it's a sequence of points which are statistically significant, but it can be more complex, can be a sequence of points or spread over time, so it's a spatial temporal cluster. Or it could even be even more general, so it could be a space, time, and frequency cluster, in which you will have like a 3D tensor where you have space, time, and frequencies. This is summarized, and this is now allowed by cluster-based statistics, in which you don't do the statistics at the level of the single value, say the single time point, but at the level of clusters. And so there are a set of tools in which they calculate the p-value associated to a given cluster that passes a given threshold. So here on the left, you have what we call the test-wise so test level methods, which allow you to calculate a p-value, which is then corrected for multiple comparisons at the level of each time point, say, using, say, FDR correction, Bonferroni corrections, and so on. And here on the right, there is a second set of statistical tools that do not work at a single time point or single test-wise level, but they work at the cluster-based level. And so the output will be for a given cluster in time or space-time, a p-value which is corrected for multiple comparison. And so you will have an output which will be common both for the different types of analysis. This type of workflow is general, meaning that it can be used both to analyze the function, the relation between, say, imagine that X is the brain activity of a single brain region and Y is the experimental condition. So this workflow, if X is a single node activity, will produce a map of p-values showing the brain regions which are statistically correlated with the experimental condition. On the other hand, X could be more general. So X would be like a network level activity. So X could be the functional connectivity between two brain regions, and Y would be, again, the experimental condition. And so at the end of the analysis, you will have a map of the links which are statistically significant, which are significantly encoding the experimental condition. And so you will be able to compare both the local activity, local encoding, and encoding at the network level. I hope I didn't go too fast. But so if you want, and I think I would suggest you to try to go through this like bottom-up approach using workflows as a start, because they combine the different steps in a single kind of in 5 lines. You will be able to do all the analysis. So these workflows are stored into the directory core/workflow, and it contains a set of pipelines which are helpful for you to go from the data to the output p-values. So the starting point, again, is your brain data. It can be LFP, spiking activity, even power, like time frequency power maps that can be organized in single subject, multiple subject, or even single subject with multiple session or multiple brain regions. And then a task variable, which is related to your experimental conditions. So the stimulus type, or experimental condition, or it can be a continuous variable such as behavioral models such as prediction error, surprise level, or reaction times, and so on. So the first step in your analysis, you will see that will be to create and manipulate your data to make it readable by the workflow that we implement in FRIT. So the way that you need to organize your data is through what we call the .dataset class. So the dataset FI, so for electrophysiology, is a class which takes as an input the brain data, the task condition, and then it checks for different, if the data is properly formatted, and it converts it so they can be read by the workflows. So this function here is probably the only moment in which you will need some kind of, it would be kind of error prone, because you will need to manipulate your data and make it readable and format it in a way that FRIT recognize it. But then once you will do this, this will be the input of the workflows for, say, for mutual information, okay? So there is a workflow which is called WFMI, so workflow for mutual information, which allows you to measure the effect size, say, of each brain region, linking the brain data to the experimental condition. And it will provide a measure of the effect size, so which is this first initial part, so the effect size, okay, per subject or across subject or across sessions, depending on how you organize, what data you put in. So the red box is exactly this first square at the beginning. So depending on the type of analysis you may want to do, the red block, so the red workflow, will create the permuted and the effect size of your data. The second step is the blue box, which is the second part of the analysis that you see here, which will allow you to do the statistical analysis, and it's called WF-Stats, so workflow for statistics. And the goal of this workflow will be to calculate the effect size from the chance level and to correct for multiple comparison, so that the end result will be from one side the effect size, so the amount of, so the t-values or the information values between the brain regions in a dynamic manner across the population and so on, in addition with the significance level, so the p-value which will contain the p-value corrected for multiple comparison. Okay, so I'm getting to the end. So this is a recap. So this is the website of the toolbox. There are some examples. I suggest you to have a look in the next day. So before going into the tutorial in English, so into the in the school in English, I suggest you to have a look at the in addition to the theory, also to the examples that you have on the website that allows you to do, say, to create a multi-subject data set. So this is probably the first initial blocking point. Then there are some examples of how to create group level statistics on the measures of information, some very simple connectivity and information transfer analysis. Then there are some examples on the statistics and so on. And if you want to try this out, you can do some, there is some codes which allows you to simulate some data. So we have some very simple network level simulations based most of the time on autoregressive multivariate or autoregressive models, multivariate autoregressive models, which allow you to create data sets of either transient activity, which would be high gamma activity or multi-unit activity, and so on, or oscillatory activity such as oscillating network with a directionality, say in a given frequency band, say in a 40 Hertz band. And so with the simulations, you may want to simulate some data and then analyze them with the tools that they are shown in these examples. To wrap up, so what can Freak do for you? So it may allow you to do network-based analysis and also region-based analysis, so RI-based analysis or LFP-based analysis, single electrode analysis in the same kind of framework. Contains a different set of measures of functional connectivity that can be used for either single trial, also dynamics or time resolved functional connectivity, such as mission information, range of causality, can be directed and undirected. Can be used to both atlas-based and channel-level functional connectivity. So this is a detail, but it's not going to be a detail if you have the data which is sparse. So imagine that in one case, what I called atlas-based analysis is, say, imagine that you have for 20 subjects the same spatial resolution, spatial atlas. So imagine you have 100 brain regions across 20 subjects and so on. This is what I call atlas-based analysis. But in case you have, say, LFP or intracranial EEG data analysis, you may have channels which you may have subjects which are different, so with a sparse data set. And we actually developed the tools so that it can be applied both to whole brain and sparse data, such as LFP, multi-unit to LFP data analysis. And then, as I showed you, can be used to do single participant or group level statistics. Yeah, so we linked it's integrating into the eBrains infrastructure if you are using it, so it can be used also from outside. In terms of link with different toolbox, the input data can be either NumPy array as always, but also you can read also MNE formats, which is what we use in the pre-processing. And also, I mean, we are trying also to probably, this is something that is still missing, try to link it with Neo and Elephant, but the input data can be pretty general. And as a final remark, so this is the repository of a tutorial in which you can start to work depending on your schedule and also depending on what you want to do during the week. But it contains, it's called Cooking Frit. So it contains a set of tutorials and a data set with Jupyter notebooks that allows you to go through a data set from the initial formatting part, creating data set, to the final steps that allows you to calculate the statistics and so on. And I think I can stop here.
Great. Andrea, thank you so much for this wonderful deep dive into the world of functional connectivity estimation. And also for the practical parts. So I can't wait to try it out. As always, Andrea just already mentioned the tutorials that let you sort of explore many of the methods that Andrea was talking about today in detail. Yes, and during the in-person week, you will then have all the time in the world to ask Andrea questions that are still open, discuss with him analysis approaches that you're interested in some of the data sets that we have available. Yeah, and talk to him about the project. So again, Andrea, thank you so much for being here and for sharing all of this wealth of information with us. I think this was super interesting. And yes, I'm looking forward to seeing you at the course.
Thank you for organizing. I think it's going to be very, very useful also for next year's students and so on. And for all the students, good luck. Don't hesitate to contact us to have some more information, and see you soon. Bye-bye.
Great. Thanks, and bye-bye, Andrea.
Bye-bye.